cancer has been present throughout recorded history. And some of the earliest evidence of cancer is found among fossilized bone tumors, human mummies in ancient Egypt, and ancient manuscripts. The oldest description of cancer was discovered in Egypt and dates back to about 3000 BC. It's called the Edwin Smith Papyrus and is a copy of part of an ancient Egyptian textbook on trauma surgery. It describes 8 cases of tumours or ulcers of the breast that were removed by cauterization with a tool called the fire drill. The writing says about the disease, there is no treatment. However, in the 16th and 17th centuries, it became more acceptable for doctors to dissect bodies to discover the cause of death. And the early 20th century saw great strides made in understanding the structures, functions, and chemistry of living organisms. Cancer research in cell culture, chemical carcinogens, diagnostic techniques, and chemotherapy firmly established oncology as science. Today, cancer research continues to evolve, and scientists have learned more about cancer in the last 2 decades than had been learned in all the centuries preceding. The growth in our knowledge of cancer biology has led to remarkable progress in cancer prevention, early detection, and treatment. Here are some global statistics on cancer. In 2023, it is projected that there will be 1,958,310 new cancer cases and 609,820 cancer deaths in the United States. Globally, there were an estimated 20 million new cases of cancer and 10 million deaths from cancer in 2023. The cancer burden is expected to increase by approximately 60 over the next 2 decades, further straining health systems, people, and communities. What are the different types of cancer? Cancer can be grouped by body location or system. Here are some common types: colon and rectal cancer, endometrial cancer, kidney cancer, leukemia, liver cancer, lung cancer, melanoma, non-Hodgkin lymphoma, pancreatic cancer, and many more. How does cancer start, and how does it develop? Cancer development is a complex process that involves multiple steps. The first step is initiation, in which a change in a cell's genetic material, a mutation, primes the cell to become cancerous. This change in the cell's genetic material may occur spontaneously as a random event or due to a gene mutation, or be brought on by an external exposure to a substance that causes cancer. The second and final step in the development of cancer is promotion, which allows a cell that has undergone initiation to become cancerous. Cancer can grow directly into or invade surrounding tissue or spread to tissues or organs, nearby or distant. The development of cancer takes place in a multi-step process, and as the cells become more abnormal, they gain new capabilities, such as the ability to release growth factors and digestive enzymes. The cells continue to divide, impacting nearby normal cells, often reducing the function of the affected organ. Why is there no cure for cancer? There is no single cure for cancer due to its complex nature and the fact that it is not a single disease with a single cause. Here are some key reasons why there is no cure for cancer. Cancer is not just one disease. Cancer is an umbrella term for over 200 distinct diseases, each with its own unique characteristics and challenges. This makes it difficult to develop a single cure that can effectively treat all types of cancer. treatments can eventually stop working, cancer cells can acquire genetic mutations over time, leading to changes in their behavior and the development of resistance to treatment. This can make it challenging to find long-term effective treatments for cancer. Limited understanding of the disease. Despite significant progress in cancer research, there is still much to learn about the underlying mechanisms of the disease. Scientists do not yet have a complete understanding of all the factors that contribute to the development and progression of cancer, hindering the development of targeted and effective treatments. Cancer cells can remain in the body. Even after successful treatment, some cancer cells may remain in the body and have the potential to grow, divide, and form new tumors. This is why doctors often avoid using the term cure when discussing cancer, as there is always a risk of the disease returning. Although there is no single cure for cancer, significant progress has been made in improving survival rates and developing more effective treatments. Ongoing research and a better understanding of the disease will continue to drive advancements in cancer prevention, diagnosis, and treatment. What are the common cancer treatments? Surgery, a procedure in which a surgeon removes cancerous cells or tumors from the body. Chemotherapy, the use of drugs to kill cancer cells or shrink tumors. It can be administered orally, intravenously, or topically. Immunotherapy, a treatment that helps the immune system fight cancer by using substances made by the body or in a laboratory to boost, direct, or restore the body's natural defenses against cancer. Radiation therapy, the use of high-energy rays to kill cancer cells or shrink tumors. Here are the pros and cons of each cancer treatment. For surgery, it can be curative for some early-stage cancers. It provides immediate results by physically removing the tumor, and it can be used for staging and diagnosing cancer. However, it is an invasive procedure with potential risks and complications, and it may not be suitable for all patients, depending on the location and stage of the cancer. For chemotherapy, it can be used to treat various types of cancer. It can be administered orally, intravenously, or topically, and it can be used in combination with other treatments for better results. However, it can cause side effects such as hair loss, nausea, and fatigue, and it may not be effective for all types of cancer. For immunotherapy, it can help the immune system fight cancer. It can be effective for some types of cancer that do not respond well to other treatments. However, it can cause side effects such as fatigue, skin rashes, and diarrhea, and it may not be effective for all types of cancer. For radiation therapy, it can be used as the only treatment for some cancers or in combination with other treatments. It is highly effective at killing cancer cells, and the treatment is painless. However, the radiation can affect surrounding healthy tissue, and it may cause side effects such as fatigue, skin changes, and hair loss. We focus our discussion in radiation therapy. What is radiation therapy? Radiation therapy, also known as radiotherapy, is a common cancer treatment that uses high-energy particles or waves, such as X-rays, gamma rays, electron beams, or protons, to destroy or damage cancer cells. It is a local treatment, meaning it is usually aimed at and affects only the part of the body needing treatment, while minimizing harm to nearby healthy cells. Advantages of utilizing radiation therapy. It is an effective treatment. Radiation therapy is a reliable and effective cancer treatment that has been used for over a century. It allows precise targeting. Modern methods of radiation therapy can aim beams directly at the cancer while protecting healthy tissues from high doses of radiation. And it can be used as a combination therapy, radiation therapy can be used in combination with other cancer treatments, such as surgery, chemotherapy, and immunotherapy, to improve the chances of successful treatment. However, the disadvantages of using radiation therapy include side effects. Radiation therapy can cause side effects, such as fatigue, skin changes, and hair loss, although these side effects are usually temporary and can be managed with proper care. And limited reach. Most types of radiation therapy do not reach all parts of the body, so they are not helpful in treating cancer that has spread to many places within the body. Here are the types of radiation therapy. First is the external beam radiation therapy, or ERD. This is the most common type of radiation therapy, where beams of radiation are precisely aimed at the cancer using a machine that moves around the body. Second is the internal radiation therapy, or brachytherapy. This type of radiation therapy involves placing a radioactive source, such as seeds, wires, or tubes, directly into or near the tumor. We focus our discussion on the external beam radiation therapy. The advantages of using external beam radiation therapy include precise targeting. Modern methods of radiation therapy aim beams directly at the cancer while protecting healthy tissues from high doses of radiation. External beam radiation therapy is non-invasive. The treatment is delivered externally, so there is no need for surgery or incisions. It is an outpatient procedure. Thus, patients can receive treatment and return home the same day. And lastly, it can be used as a primary treatment or in combination with other treatments, such as surgery or chemotherapy. However, external beam radiation therapy has its own disadvantages, which include side effects. Healthy cells may be damaged along with cancer cells during radiation therapy, leading to side effects such as fatigue, skin changes, and hair loss. And external beam radiation therapy requires a long treatment duration. It is typically given in multiple sessions over several weeks. Before proceeding further, let us answer this question first. What is radiation? Radiation is energy that moves from one place to another in the form of waves or particles. It can be categorized as either ionizing or non-ionizing, depending on its energy level and its ability to ionize atoms and molecules. Ionizing radiation is a type of radiation which carries enough energy to ionize atoms and molecules, causing changes at the atomic level when interacting with matter, including living organisms. Examples of ionizing radiation include alpha particles, beta particles, gamma rays, and neutrons. Non-ionizing radiation, on the other hand, is a type of radiation that has lower energy and does not have enough energy to ionize atoms or molecules. Examples of non-ionizing radiation include radio waves, microwaves, and visible light. When ionizing radiation interacts with cells, several things can happen. The radiation could pass through the cell without damaging the DNA. The radiation could damage the cell's DNA, but the DNA repairs itself. The radiation could prevent the DNA from replicating correctly, or the radiation could damage the DNA so badly that the cell dies. The specific cellular response to radiation depends on factors such as the cell type, the type and dose of radiation, and the ability of the cell to repair DNA damage. While the goal of radiation therapy in cancer treatment is to selectively damage or kill cancer cells, it can also affect nearby healthy cells, leading to potential side effects. Careful planning and precise delivery of radiation are essential to maximize the therapeutic effect on cancer cells while minimizing harm to normal tissues. And you, dear viewers, might wonder, where do these high-energy particles or radiation beams used in radiation therapy come from? The most common devices used to generate radiation beams used in external radiation therapy are the linear accelerators or LINACs. Linear accelerators generate high-energy X-ray beams or electrons for treatment. They work by accelerating electrons to nearly the speed of light and then directing them at a target, usually made of tungsten, which produces X-rays through a process called Bremsstrahlung radiation. They can produce a range of energies and shapes for customized treatment plans. To further visualize the radiation generation, let us watch this visualization provided by Elekta, a global company that develops and produces radiation therapy equipment.
How the linear accelerator works. All body cells can be damaged or killed by radiation, but tumor cells are more sensitive to radiation than normal cells. Radiation therapy uses this principle to damage beyond repair or kill the abnormal cancer cells in a tumor. Successful radiation therapy depends on the ability of the linear accelerator, or LINAC, to deliver a tumoricidal dose of radiation to the tumor, while ensuring minimal radiation of normal tissue. The LINAC accurately produces, monitors, controls, and conforms the radiation beam to the planned target. Beam generation. Radio frequency waves are pulsed into the waveguide by the magnetron. This is synchronized with the injection of electrons into the waveguide by the electron gun. The radio frequency waves accelerate the electrons along the waveguide to a speed approaching the speed of light. The X-ray beam is created when the electrons hit and interact with a tungsten target at the opposite end. The magnetron controls the power and frequency of the radio frequency waves, which determine the energy of the X-rays produced. The digital accelerator uses a diode-type electron gun situated at the end of the waveguide. The electrons are produced by heating the tungsten filament within the cathode and are then injected into the waveguide. The number of electrons injected is controlled by the temperature of the filament. The electrons are accelerated along the waveguide toward the target. The waveguide contains a series of copper cells. Small holes, or irises, between these copper cells allow the electrons to travel along the waveguide and help to focus the beam. A vacuum is created to ensure that the electron beam is not impeded by other particles. The path of the negatively charged electron beam is controlled by two sets of quadrupole magnets, called steering coils, that surround the waveguide. An additional two sets of focusing coils help to further define the electron beam so that it is very fine, with a diameter similar to that of a pinhead when it hits the target. The entire system is cooled by water. The electrons exit the waveguide and enter the flight tube, where the beam is redirected towards the target. The electrons travel along a slalom path within the flight tube. 3 pairs of magnets on either side of the flight tube cause the electron beam to bend through the turns of the slalom. This process not only positions the beam to strike the target, but it also further focuses the beam to a diameter of 1 millimeter. The design of the magnets enables them to focus electrons of slightly different energies onto the same point on the target. This is called achromatic behavior. This slalom bending is unique to Elekta linear accelerators. It helps to minimize the size of the machine and ensures that its isocenter remains low, which is important for patient setup. The high-energy electrons hit a small tungsten target where the electron energy is converted into photons, or X-rays. The high-energy photons emerge from the target in a variety of directions. The primary collimator only allows forward-traveling X-rays to pass through, creating a cone-shaped beam.
From this video, we have already tackled some historical, statistical, and physical information about cancer. We also discussed some cancer treatments, including radiation therapy and linear accelerators, as well as a basic discussion on radiation. On our next video, we'll be presenting the specifics of external beam radiation therapy. more specifically the particle radiation therapy. We will also tackle some basic physics of the interaction of radiation through matter and, ultimately, the current challenges and gaps in this research area in medical physics.